nucleic acid. Nucleic acids are the biopolymers, which means that they are made up of many monomers that are bound together by the covalent bond, and the macromolecules, which suggest that these are the large complex molecules which are essential to all forms of life that exist. There are two main classes of nucleic acids. They are DNA and the RNA. DNA, it is a double stranded, and whereas RNA is a single stranded. mRNA is also known as messenger RNA, and the rRNA is known as ribosomal RNA, and the tRNA is known as transfer RNA. These are the three classes of RNAs. As we all know, the nucleic acids are made up of many monomer units. These units are known as nucleotides. What is nucleotide? Nucleotide, it is a molecule which is composed of three major components. Those are, first one is the nitrogen base, second sugar, and the third phosphate. Nitrogen bases are nothing but these are the organic molecules which contain nitrogens in their rings. And which are the four nitrogen bases we have studied in the previous class? These are adenine, guanine, uracil, and the thymine, whereas sugar is commonly known as 5-carbon sugar. If this 5-carbon sugar is present in the DNA, it is known as deoxyribose sugar. Similarly, if this 5-carbon sugar is present in the RNA, it is known as ribose. Whereas the phosphate, if there is a single phosphate, it is known as monophosphate. If it is 2 phosphate, then it is called as diphosphate. If there are 3 phosphate, then they are known as triphosphate. Altogether, this forms the nucleotide. Here, as you can observe, I have written these three technical terms: the first nucleobase, second nucleoside, and the third nucleotide. All these terms are necessary for better understanding of the nucleic acid formation. If there is a presence of only nitrogen base in a structure, then such structures are called nucleobase. For example, one of the nitrogen base is the adenine. If this adenine is bound along with the 5-carbon sugar, such structures are called nucleoside. And the example, the adenine is now get converted into adenosine. Further, moving towards the nucleotide, as I have mentioned previously, nucleotide is the polymer which is made up of three major components. These are nitrogen base, 5-carbon sugar, phosphate group. Now this adenosine is now known as adenosine triphosphate, and hence this structure is known as nucleotide. Now let us understand the arrangement of all these components. First, always the sugar molecule is placed centrally, and the phosphate group and the nitrogen base are present on the either side of the sugar molecule. Now let us understand the arrangement of the nitrogen base, sugar molecule, and the phosphate by the example of DNA. Here it is the example of the DNA at the micro level structure. As you can see, let us concentrate on this portion. As for now, this is the sugar molecule. It is written that it is deoxyribose. As I have mentioned earlier, that the 5-carbon sugar present in the DNA is known as deoxyribose, and the 5-carbon sugar present in the RNA is known as ribose sugar. So since it is clear, now this sugar molecule is centrally placed, and the phosphate and the nitrogen base, that is the thymine, are placed on the either side of the sugar molecule. So moving towards the bond between the adenine and thymine, they are the hydrogen bonds, and there are 2 hydrogen bonds between the adenine and thymine, and there are 3 hydrogen bonds between the guanine and cytosol. As you can see, there are 3 hydrogen bonds, and the bond between the nitrogen base and the sugar molecule is known as glycosidic bond, and the bond between the sugar molecule and the phosphorus or the phosphate group is known as phosphodiester bond. Now let us further know more about the purines and the pyrimidines. What are purines? Purines are the heterocyclic aromatic compound that consist of 2 carbon nitrogen ring, and whereas pyrimidine have only one carbon nitrogen ring. Let us view in the example. See, it is the purine, as I mentioned. Example for the purines is the adenine, and this is the structure of adenine. Here you can see the two rings, and whereas in the pyrimidine it is the presence of only one ring. So that this makes a huge difference between the purines and the pyrimidines. Now here are the nitrogen molecule, and here there will be presence of carbon. Wherever I have not written, there is a presence of carbon, and this makes the difference between the purines and the pyrimidine.